Hi, everybody. Welcome to the beginning of 21B. Today, we're going to begin with our review of 21A because this is a two-part series. The second half of it is going to be part of the comprehensive part. So since we didn't have the opportunity to go through A together, I am going to review with you guys some main topics and then kind of narrow it down from there and help you guys understand what it is that you're going to be tested on for the comprehensive part of this class. So I'm going to go through this pretty fast, mainly because you should have just learned it. That's assuming you just took 21A. And then the second part of that is it's all already material that you guys have covered extensively. So if you guys do need to slow it down, feel free to do that or go over it a couple more times. But for everybody in general, I'm going to go through this fairly quickly. So this is going to be compartmentation, so cells and tissues. The functional compartments of the body, basically separating the inside from the outsides, right? What are the advantages and disadvantages? You're separating with the biochemical processes that can conflict. So protein synthesis is occurs in one area, and then protein degradation. So basically the making up and the breaking down of proteins occur in different areas, which is a really good thing because you don't want to accidentally have them occur in the same area and try to make them and break them in the same area because your body would get really confused and be really inefficient. Another advantage, they allow for lysozymes to exist in the cells. Lysozymes, hopefully you guys remember, are going to help with getting rid of like damaged cells. So that's really helpful for your body to make sure that you have different areas that are going to clean up. Disadvantages, there's barriers, so it makes things more difficult to go from one place to another, but we have special, I'm sure you guys remember, transporters for this. Definitely go through this quick. This is just different body cavities. So you've got cranial, thoracic, abdominal, and then pelvic or abdominal pelvic together. This is the plural. This is the parietal. Front is anterior and posterior, or you could also call that ventral and dorsal. A lumen is a hollowing of a area, so it's an opening. So there's lumens of hollow organs. There's hollow organs of the heart, the lungs, the blood vessels, and the intestines. Lumens for some organs are not in the internal environment; they can be external. The fluid is going to be ECF, ICF, which is basically outside of the cell and inside of the cell. A further division, so ECF is going to be like blood and IF, and then the cells are going to have the intracellular fluid on the inside, which is like the cytosol. Compartments, so there's layers, obviously, because your organs are really important. You need lots of protection. So you have the visceral and the parietal membranes, and then sometimes you'll have those sitting inside of different sacs, if you will, and then they have tissues and cells, and then phospholipid bilayers that protect them all. The cell membrane is important. It's for isolation, basically from keeping the outside stuff out and the inside stuff in, as well as for support. They're made up of lipids and proteins, and they are what we would call the fluid mosaic model, which is the phospholipid bilayer, which is, so phospholipid is what it's made of, right? These are phospholipids, and then bi just means two layers. So you've got one here and one here. You've got the head and the tail. The head is going to be hydrophilic. The tail is hydrophobic. So they're pointed towards the inside, which means they don't interact with the water. And then in the membrane, you have things like glycoproteins and protein carriers and cholesterol and things that help with movement or communication. So for example, protein carriers are going to be, this is a protein embedded in the plasma membrane that's going to help move things from one side to the other. Cholesterol is going to help because it's kind of sticky. It's going to help things adhere. Glycoproteins help with the structure, and then you also have peripheral proteins and then integral proteins. So basically it just has to do with where the protein sits. Same thing of what I just said. This has to do with the formation of the phospholipids. This is a bilayer, this is a missile, and this is the liposome. We'll talk a little bit more about missils when we get to the digestive system. Cholesterol is really important. Obviously, it has good benefits to your body, like it helps things stick. But then also, if it clumps up too much, it can be really bad, obviously, because it can start to clump to the point where it's inhibiting something like your blood from moving. Proteins, again, integral and peripheral. They are mainly for transportation. They help increase enzymatic activity. They are receptors for things to bind. They help with intracellular joining or adhesion, and then cell to cell recognition, which is just that the cells identify each other and can communicate. The cell and the organelles, right? So it's the basic unit and structure and function of the body, really diverse, and then the parts to it. So hopefully you guys remember, right? The cytoplasm, the nucleus, the cell membrane, just in general, right? Cell membrane, cytoplasm, the nucleus. So for division, content, just to keep, hopefully you guys talked about tonicity, to keep the tonicity isotonic, and then also the nucleus is to house the DNA. And then you get into all of the structures. So ribosomes for protein synthesis, microtubules are for mitosis, microfilaments are going to be for structure, cilia and flagella. Cilia is going to be to increase surface area, flagella is going to be for movement, cytoskeeleton is for shape. Let's see. Okay, so you would just go through and make sure you remember those. These are really, really, really basic. Honestly, so basic, you guys, that I'm not even going to cover them in the review portion of the exam. Obviously, mitochondria is the ATP powerhouse. There's two types of ER, rough and smooth. Rough is for protein synthesis, smooth is for lipid synthesis. Golgi is the packaging and processing center. Lysosomes are to break down anything bad. Secretory vesicles are to transport things. And then we get into apoptosis of cells. So apoptosis is a process by where you would have basically spontaneous cell death. So this is the normal cell that is going to be changing its shape, fragment, and then kind of randomly bud off into these little buds or undergo apoptosis, the little bodies there. So this process normally occurs normally to allow for cellular turnover, right? Your all of your cells kind of have a shelf life, if you will, and you want to make sure that everything is functioning optimally. So when anything is old, dead, or dying, your body's going to get rid of it and make something new. Peroxisomes are going to help with, so enzymes are going to degrade fatty acids and then any toxic foreign molecules. They also help break down and generate hydrogen peroxide. These are the peroxisomes. Nucleus is, I feel like one of those really redundant ones, but again, we can take it a step further here. So nucleus is where you house the DNA, which obviously is really important because it's your genetic makeup, but then you also can go into genomes and proteomes from there. So a genome is going to refer to all genes in a particular individual. It's your genetic code. So when we say code, what are we talking about? We're talking about polypeptide chains of nucleic acids. So if you guys remember, that was the ATCG, which is the adenine, thymine, cytosine, and guanine. So adenine and thymine bond together, cytosine and guanine bond together. When those hydrogen bond together and they make up the double helical form, that is going to be your genetic code. A proteome is going to refer to all of the polypeptides produced by that genome. So in this case, you have RNA that's coded by the gene. It can be spliced into different ways. So if you guys remember talking about it, it gets spliced and separated depending upon the introns and the exons. The introns are basically garbage and you get rid of them, and the exons get spliced together. So that's the basically usable code that your body's going to just naturally put back together. So polypeptide chains are going to be associated with other chains, and they are then going to be yielded into proteins. So they're going to be translated into proteins. I'll review with you guys that in just a bit. Carbohydrates and lipids are going to be added then to alter the functioning. So for example, it's a post-translational modification, which again, RNA into proteins is translation. So after that happens, you can have like a methylation or a phosphorylation or a cleavage occur, and you're actually going to change the way that it expresses itself because of that change. So here's a non-dividing cell. If you look on the inside of it, Or you can see the chromatin. The chromatin, if you guys remember, is basically it's like a ball of yarn, but it's still your DNA. So it's just not wrapped perfectly. So when you wrap it perfectly, right, you condense it into chromosomes, you put the strands around histone proteins, and they kind of wrap themselves around those proteins, and then they eventually become this nice double helix structure. At this point, you would then be able to call it chromosomes when they are nicely raveled like that. See, this is the chromosome. But again, you can break it back down and pull it all back apart, and then you would eventually be able to get the backbone, which you guys should remember is the sugar ring, which is this. It's a pentose sugarine, which means 5-carbon sugar, and then the phosphate molecule with that, basically that arm, right, the side chain is going to be the nucleic acid. So again, it's either adenine, thymine, cytosine, or guanine. Each one of those would be different for those purple ones. So after, right, post-translational modification, you could have the condensed chromosome. You can undergo an acetylation, which is going to produce a more open structure. It unravels it. Transcription factors then can attach to the chromatin, activate the genes, produce the RNA, and then have something like a de-acetylation, which means you're getting rid of it. You're taking that chromatin and you're silencing the genetic transcription factor now. So you have the ability to play essentially with the DNA and the genetic code as you want, maybe in a lab environment, or sometimes it just happens naturally in your body. So here would be the kind of summary of all of this again, right? So summary of DNA replication, you guys, the first thing that has to happen is you have a helicase enzyme. The helicase is going to come in. It basically like, this is a zipper, right? Unzips the zipper. Then you have a single strand that is going to then have a complementary strand come attached to it. Now you've got 2 different strands, right? So now you've basically doubled it. So the leading strand is going to be synthesized continuously from the 5' to the 3' end, and then you're going to do the 3' to the 5' end, and basically make it a complementary strand. In this case, on this side, you have what we would call the lagging strand. It's synthesized discontinually. So this is, you guys can see that zipper right there, the light blue one is all the way through. The lagging strand is discontinuous. That means that you have a primase enzyme that's going to synthesize the RNA primer. It extends to the DNA polymerase, which is another enzyme, to form what we call Okazaki fragments. The Okazaki fragments would be formed on the lagging strand, after which point you're going to have an RNA primer placed on the DNA. Then the DNA ligase is going to start joining those fragments together, right? You can see that the joining of the fragments will come together, and the strand is going to then grow in length. So the Okazaki fragments essentially aren't able to be seen anymore, and you're going to now join these end to end. Here's a really good representation for you guys of adenine, thymine. So you always see red and blue together, right? Red and blue, red and blue, red and blue, and then cytosine and guanine. If you guys remember back to like a general bio course, it's because adenine and thymine hydrogen bond 2 times, and cytosine and guanine hydrogen bond 3 times. Obviously, you don't want something left unbonded because then it's going to have a charge. It's not going to be happy. So you always want a T to bond and then CG. Telomerases. So with telomers and telomerases, with these guys, you have, so telomerases are going to give you an idea of your ability to divide. So the decrease of your cell's ability to divide is going to be an indicator for you of aging. So sometimes you can do different tests, right, on your genes and find out how well you're aging in terms of genetically, not necessarily physically. So the aging is going to be related to the loss of the DNA sequences, which you guys see here, which are going to be the ends of those chromosomes. Those regions are going to be called the telomers, right? So the telomere is going to be the end of it. The telomers serve as a cap, and the caps are going to prevent enzymes from mistaking the normal ends for the broken DNA. We have DNA pull, or DNA polymerase doesn't fully copy the DNA at the end regions. So each time a chromosome replicates, it loses about 50 to 100 base pairs in its telomere. When you guys have things like germinal cells, the germinal cells are going to give rise to the gametes. They can divide indefinitely due to the enzyme that we just talked about, telomerase. That enzyme is then going to duplicate the telomere DNA, and this is also one that we find in, let me see if I can go back. Yes, one that we find in cancer cells. So if your cells are aging faster than they really should, or something went wrong and something mutated, the telomeres wouldn't have the right, there it is, the right age for your physical age. And in this case, the ends start to kind of unravel and tell your body to do something differently. So telomerase is found in cancer cells, so specifically hematopoietic stem cells and other stem cells that are going to continually divide, that you guys find in your body all the time doing work. How that happens, right? How they divide is through the cell cycle. So if you guys remember a cell cycle, you've got interphase and you've got G1, S, and G2, and then you've got mitosis, and then at the end you've got cytokinesis. So interphase is basically growth, DNA replication, and more growth. And then you've got mitosis, which just in the, okay, this one's correct. In the last couple years, there's been an extra stage added in of mitosis. So mitosis is now, you can either call it early prophase or just call it prophase. Then you've got late prophase. This is also called prometaphase. And then you've got metaphase, anaphase, and telophase. And most of the books that I work with, it's prophase, prometaphase, metaphase, anaphase, telophase. So literally what they did is they just combined prophase and metaphase and put it in the middle and called it prometaphase. So you have basically the beginning, right? You've got the nuclear envelope dissolves, the spindle fibers form, and the what else is there? 1, 2, nuclear envelope, spindle fibers, and then the chromatin, which is the like jumbled up ball of yarn, that's the DNA, is going to condense into chromosomes. That's the big one here. Then in prometaphase, you're going to have the spindle fibers attached to the centromere, which is the waist of the chromosome. Metaphase, the chromosomes align along the equator. Anaphase, the chromosomes divide into daughter chromatids. And then in telophase, you're going to have basically the reversal of prophase with a cleavage. So the cleavage is telling you it's the beginning of the division. And then you're also going to have like the nuclear envelope reform, the spindle fibers disappear, and then the nuclear envelope reforms. The most identifiable feature, obviously, is the cleavage furrow that's developing. That will lead you into cytokinesis, which is the complete division of the daughter cells, which are going to be genetically identical to the parent. Now, mitosis is going to be driven by what we call cyclins. Cyclins is basically a protein. So cyclin D is a protein that is going to help activate the different kinases that are going to help motivate the production of all the stuff that you need. Oh man, I missed it again. There it is. All the stuff that you need to form in G1, S, well, not S, but then G2 as well. So if you have cyclin D and it's activating the cyclin to help the cells move quickly through G1, that also then can be correlated to cancer. So if you have overactivity of this gene that codes for cyclin D, it's going to cause or might cause the uncontrolled cellular division, which is basically what we would also call cancer. So if you have overexpression of cyclin D1, right, that would be like the gene. It's also going to be known to be linked with cancer, specifically breast cancer or esophageal cancer. So these were the cyclins that I was talking about. It's a very detailed process, obviously, that we're not going to talk about today, but it does happen. Just a tiny little bit about oncogenes. So oncogenes are going to be genes that contribute to cancer by basically starting uncontrolled cell division. Oncogenes then are going to be mutated mutated forms of normal genes in that function in healthy cells. And then we also have things that are called proto-oncogenes, which are going to code for proteins, products that regulate cell growth, division, and adhesion. Any mutation of any of those are going to lead to oncogenes and then basically result in activity that you can't control anymore, which again, we would call or term cancer. So we have what we call tumor suppressor genes as well. So if you're looking at this signaling pathway, we're going to be talking about the P53. So tumor suppressor genes are going to be types of genes that are going to hinder cancer development. So basically stop it, which is a really good thing for our body. So one of them is going to be the P53. So you guys see the P53 all over the place over here. So the P53 is going to be a gene that indirectly blocks the ability of the cyclins to stimulate cell division. So it acts as a transcription factor, right? Transcription is DNA and RNA, to prompt the expression of a P21, which is going to inactivate the cyclin dependency. So if P53 is active when the DNA is damaged by something like radiation or chemicals like asbestos, or sometimes even just cellular stress, the cell cycle is going to be slowed, which gives your body more time to repair the damaged areas or trigger what we would call apoptosis, which means that your body's going to realize that, hey, this is wrong. I need to fix it, and either kill it because you can't fix it, or just kill it because your body's realizing that it's wrong. So examples here would be like skin cancer, squamous or basal cell cancer, right? These are going to be cancers that are believed to involve the mutations of the P53. So obviously, you guys, if you have a P53 gene that is mutated and you can't control the rate at which the transcription factors are going through, you're more likely to develop cancer. So in laboratory studies with mice, the P53 gene has been artificially inactivated, and it's called a knockout gene. Basically, they knock out the genes. The mice can be created with a targeted gene. So there's been a lot of research in this area to help us, obviously, prevent more cancer from being fatal. We also have what we call MMPs. So MMPs are going to be matrix metalloproteinases. It's basically an enzyme, right? ASE is an enzyme, which is going to use zinc as a cofactor. In this case, they break down the extracellular matrix, and cancer cells produce the MMP. Then they use it to metastasize. So we can also see destruction of other tissues like cartilage, in arthritis from the MMPs in this particular case. Okay, intrinsic and extrinsic apoptosis. So apoptosis again is cell death. You can undergo cell death obviously in a couple of different ways. Intrinsic would be inside of the cell, extrinsic would be outside of the cell. I don't think, let me see. Yeah, we don't need to go into that too far in depth. Just know that so let me go back one. So just know that the extracellular molecules are going to be called death ligands that bind to the receptors. And in that case of the extrinsic, they're going to use an FAS receptor to remove the T lymphocytes, whereas the intrinsic is going to use stress signals to trigger the release of mitochondrial molecules and then trigger the next phase, which is a caspase. Next would be stem cells. Stem cells, hopefully you guys remember, right? They can, so they're undifferentiated. They can be derived into anything. So a stem cell can be made to replicate itself into a nervous cell, an epithelial cell, a blood cell, a fat cell, pretty much anything, right? So it has a very, very, very big ability. In short story real quick, oh gosh, man, I think it was 2 years ago, so 2018, there was an Army officer who got into a car accident, and she, in the car accident, she basically lost her outer ear, which is the pinna. Hopefully you guys remember, for the nervous system. When you lose the outer ear, if you guys remember, the outer ear is basically a funnel. So if you lose the funnel, Basically, if you put your hand over your ear, you would see what exactly that's like. You lose like the ability to hear really, really, really well. So her middle and inner ear were not damaged in the accident at all, but the doctors noticed that obviously when you open the external environment to the internal environment, there's a really big issue of infection just coming in rapidly. And she lost her outer ear. So what the doctors decided to do basically is just seal it up so they didn't cause her to get any infections that were unnecessary. Then what they got permission to do is they used stem cells. They used stem cells and they implanted them in her forearm. And what they did is they intentionally differentiated them into an outer ear, or the pinna. When they did that, they waited for it to grow, and she grew an outer ear on her forearm. When it was done growing fully, they grafted it. So they pulled it off her forearm and put it back on her ear. And then they opened the ear back up, and she regained full function of her ear. So stem cells can be really, really, really good, but you have to be very careful how you use them. There's obviously a whole bioethical board giving people approval to use them, and then obviously stopping people from using them in harmful ways. This is the review now of tissues. So epithelial, connective, muscle, and nervous. I'm not going to go over the whole thing because you guys covered it extensively last semester. Epithelial, if you guys remember, is either going to be found covering a surface or lining a cavity. Connective is going to connect in some way. There's different matrices of these two. So there's liquid, gel, or solid. And then muscle is for contractions and movement. Nervous is to send signals. I do recommend you guys reviewing these because we will make references to them in the second half of this, such as like the respiratory system has different types of tissues, and one that lines the trachea is the PCCE. It does that because it has the pseudostratified ciliated columnar epithelia with a cilia that's going to be able to move the mucus up and move the junk out, which got trapped in there. While the alveoli is going to be lined with simple squamous, which is going to be the thinnest layer, which is going to be helpful for the movement of oxygen and carbon dioxide. So you guys can see like really quickly why it's important to know what the type of tissue is and where it's found, because we will be going over that again this semester. If you guys don't remember, simple means one, stratified means more than one layer. Then you've got different shapes of the cells. Squamous is flat or squished, cuboidal is cubed, columnar is taller than they are wide. Glands are glands. They produce things either into or out of. The major endocrine glands we're going to talk about for the first section of this semester. Connective tissue for different purposes, but basically, so protection with maybe like adipose, movement of gases in blood. Cartilage is for joining things together, and then to, like, for example, in your arms or in your legs, it's for protection and absorbing shock. Bone is going to be protection. So just make sure you guys go through all of those one more time. Sorry, forgot those two. Muscle, 3 different types. This is ad nauseum, but just in case you don't remember, skeletal is voluntary, smooth is involuntary, cardiac is on your heart only and involuntary. Neurons, so 2 different types of nervous tissues, neurons and glial cells, which I'll cover more in the review of the nervous system. But neurons are going to be the ones that send signals, and then glial cells are going to be the ones that are for support and protection, which is really important because your nervous system has a very, very, very limited ability to repair itself. That takes us to the end of today's review. If you guys have any questions, please post it on the discussion board, email me, or just, let me know in any way. I'm happy to help you guys before we move on to the next section. Hope this helps you, and I hope to see you guys in Zoom.